all of those laws ultimately culminate in the fact that I can use Punnett squares to predict the phenotypes and genotypes of the next generation. I can do monohybrid, dihybrid crosses and use those Punnett squares as a tool to predict. So if we remember a monohybrid cross, mono means one. So we're looking at the pairing of parents and only considering one trait at a time. So if we go back to Mendel's plants, all right, we're using the mating of pea color, so a yellow pea with green pea, and using the Punnett square to predict the outcome. Now, Punnett squares only predict the probability of the offspring of a particular trait. Because your gametes get randomly fertilized, your actual outcomes can vary. The more offspring you count, the closer you'll get to the probability predicted. All right, but if I take a look at a brother and sister, all right, I could have just as easily gotten two offspring that look like this, or I could have had them look like this. So you've got to be careful. The smaller your number of offspring you're counting, the further off it's going to potentially look from the probability that you predict. All right, but the only reason I can use these Punnett squares is because of that law of segregation and independent assortment. All right, so quick brush up, how do we do the Punnett squares? You can do the tic-tac-toe board like this one, or you can just do the box like that, whichever works better for you. But if we do the tic-tac-toe board, all right, we place parent A's alleles across the top. Conventionally, that top section is usually the father's. All right, so if you know the sex, that's generally dad's information. Moms would then go along the other side, and then we pair the alleles in the middle. All right, so in this particular example, dad was heterozygous for the trait, mom was homozygous, and so we end up with a 50-50 combination of offspring with the two dominant versus recessive traits. Test crosses are an interesting way to determine the genotype of the parent when all I know is the phenotype. So remember I said back earlier when we were talking about the cows, right? The black cow can be homozygous dominant, or it could be homozygous recessive. All I know is that it's black. I don't know what its genotype is. So what we can do is do a test cross, where I mate an individual with a dominant phenotype, where I don't know what its genotype is, like the black cow, and I mate it with an individual who I know is recessive. Because recessive I can see, recessive I know they have to have two copies of that recessive trait, so I already know their genotype simply by looking at them. The red cow, for example. Red cow is recessive, so they have to carry two recessive alleles in order to show that trait. I mate that unknown dominant with the homozygous recessive that I know its genotype, and I observe the offspring. Counting the number of offspring that I have will help figure out whether or not that dominant phenotype is homozygous dominant or heterozygous. So here's an example of flower color. Here's my purple flower. I know that because that purple color is dominant, it either has to be homozygous dominant or heterozygous. So I mate it with an individual that is of the white phenotype. In this case, we know white's recessive, and I look at their offspring. If it's homozygous dominant, I'm going to get all of my offspring being purple. But if this purple flower was heterozygous, I'm going to get a 50-50 split. Okay, so in this case, if I see white flowers show up, I know that this individual had to be this. So some of the predicted ratios we can look at from test crosses, assuming that they are traditional Mendelian genetics. Here I've got a parental generation that is true breeding, so homozygous dominant purple, homozygous recessive for white. That F1, or that first filial generation, they're going to all be carriers. They're all going to be heterozygous, all right, 100% purple. How do I prove that? Well, I can take that F1 generation and self-fertilize, or take two F1 generation individuals and made them together, and I should get, assuming this is traditional Mendelian genetics, 3 dominant individuals to 1 recessive in my second filial generation, or F2 generation. And within that, I should be able to know that my genotypic ratio gives me 1 homozygous dominant, 2 heterozygous individuals, and 1 homozygous recessive.